Welcome everybody at a new Bytes of Innovation webinar. Bytes of Innovation is a webinar that's powered by Segmed, a company that provides medical imaging data to researchers. At Bytes of Innovation, we invite key opinion leaders to talk about the future of healthcare. This show is co-hosted by Dr. Aline Lutz, who is the Vice President of Real-World Evidence and Clinical Insights at Segmed, and by myself. My name is Martin Willemink, and I'm the co-founder and Chief Scientific Officer at Segmed. The concept is we invite a guest who will give a presentation, and then after that, Aline and I will moderate the Q&A session. So everybody in the audience, make sure to put your questions in the chat box so that we can discuss these after the presentation of the speaker today. And today, I'm very proud to announce that we have Dr. Sean Khozin amongst us. Sean is a physician scientist and oncologist, and he works at the intersection of cancer medicine, drug development, and AI. And he's a very busy guy. He's the Chief Executive Officer at the CEO Roundtable on Cancer and Project Datasphere. He's a founder of Fusion Bio and a research affiliate at MIT. And previously, he served as the CEO at CancerLink, which got acquired, and he was the global head of data strategy at Johnson & Johnson. He's also a founding member of the FDA Oncology Center of Excellence. And currently, he's still, on top of all of that work, he still has time to do research, and he focuses on RECIST and the total tumor burden index. He's authored more than 100 peer-reviewed publications with over 6,000 citations. And excitingly, he actually has been working with the Segmed team, and the paper that we worked on is about real-world imaging data and applications for pharma and life sciences. Got last week accepted, so soon we will announce the full paper. But for today, I'm excited to have Sean here. He's going to talk about the simulator's dilemma. There are no gold standards. Sean, the floor is yours. And share my screen.
Thank you, Martin, and thank you, Aline. It's great to be here. I'd like to thank the entire Segmed team. It's been quite a pleasure to work with you over the past few months on the research project that you mentioned. So I'm going to share my slides. So as Martin mentioned, the title of my presentation is The Simulator's Dilemma. And this topic essentially refers to something that I believe many of us developing clinical AI eventually have to reckon with. And that's the fact that in science, especially the life sciences and biomedicine, there are really no true gold standards. So when we think about our reference standards and labels for training AI, I believe that's something very important to keep in mind. Much of medical AI today is being built to produce human judgment and interpretation. And in fact, the reference standard in a lot of times is that expert annotation. And the model, therefore, learns not necessarily from ground truth, but observations made by radiologists, pathologists, and clinicians. Thus, the model inherits their biases and imprecisions. So learning from experts is actually quite important. I don't want to discount that. But in doing so, we have to recognize that we can only build tools that can be as good as the experts. And again, that is valuable. We can bring much needed efficiency to the biomedical enterprise and a lot of the workflows, especially at the point of care. But at some point, we have to ask whether those efforts are really building tools of discovery or simply just building faster horses. And I think that the real promise of AI is not necessarily to build faster horses, albeit that's important, but it's to really represent reality and our world in new ways, because that may in fact be the only path to new biological insights, better disease classification schemas, and ultimately better therapies and outcomes for individuals and patients, which is essentially the ultimate goal. So my central argument here is that when we confuse human convention, expertise, opinion, and with biological truth, we may end up limiting what clinical AI can become. And we may get better at automation and streamlining certain processes, but we may not get any better in doing science. So, as I mentioned, the main premise of my thesis is the fact that a reference standard is not a fact of nature. The existing reference standards are primarily a human construct with an error distribution. We can think of them as artifacts that have a history coming from a specific instrument, a specific moment in time, and specific people doing the best they could with the tools that they had. And this has, again, implications in how we name diseases, how we measure diseases, and how we validate our tools of diagnosis, including AI-powered diagnostics that have been surfacing in recent years. In order for me to demystify this, I'd like to give an example as a thoracic oncologist. I'd like to tell you how we measure and how we classify lung cancer. How do we know lung cancer is what it is? So something to keep in mind is that, and this is a relatively intriguing story, but I might be biased given I'm a thoracic oncologist. We call lung cancer not because of any mechanistic insights about lung cancer, but because of surgeons. In an era where surgery was the only treatment option for patients with cancer, the only way to organize cancer was anatomically, by where it sat in the body, essentially, by the organ you operated on. And the disease was essentially the site. That's what I call the scalpel classification. It's anatomical, which has been inherited to us. And at the time, in the 1800s, it was entirely reasonable. Then came the microscope, and something subtle happened. We started to describe what we saw under the lens in human language, linguistically. We started to describe our observations. So we had a lesion in an organ that we called the lung. We start to look at that lesion under the microscope, and we start to characterize the cells, starting with the size of the cells, because that was the most obvious thing to us under the microscope. So we realize that the cells are of varying sizes, but there's a cluster of cells that are quite small in certain disease types. So we called that small cell. And what about the rest? Well, the rest were actually not a very coherent group. It's always the case in many diseases, especially in oncology. So we simply started to call the other category non-small cell. We literally made up a phrase to refer to a heterogeneous population of cells that were not small, and they were non-small. And that's what the microscope gave us. And what's quite interesting is that in the 1960s, the WHO then classified that linguistic observation of a morphological phenotype observed under the microscope and articulated linguistically as a histological classification. So this has been one of the most consequential classification schemas in oncology, because everything else falls under this classification, including genomic profiling, that you may argue should actually be the starting point. So one of the things that I've been and many others have been arguing for in recent years, especially, is that we need to start to abandon anatomy when it comes to classifying all oncological diseases and start thinking about the mechanism of disease and be a little more bottom up. And in a way, this is already happening without much interference by experts. So if we look at the evolution of classifying cancer, as I mentioned, anatomy was the starting point. Then we moved into histology, and more recently, we started to interrogate the genomic underpinning of tumors. So genomic classification became the norm in many cancer subtypes. And where we're going as part of this natural evolution is multimodal characterization of disease, which is more mechanistic and would give us much better insights that where the location of the tumor is. So as I mentioned, this has been the natural evolution of cancer classification to the point that today there's a category of disease indications that are tied to, for example, immune checkpoint inhibitors that are tumor agnostic, and they're solely dependent on the genomic signature of the tumor. These are called tumor agnostic indications. So in a way, we've already abandoned anatomy unknowingly, in a way, and have become much more mechanistic when it comes to classifying diseases. So pembrolizumab, which is an immune checkpoint inhibitor, was the first therapy that was given a tissue agnostic indication for patients that have a high tumor mutational burden or are microsatellite unstable. And these are essentially genomic mechanistic markers. So anyone with those markers, regardless of where the tumor is in the body, can receive pembrolizumab. It's an FDA-approved indication. And so one way to think about this is that AI can be leveraged not just as a faster classifier that can again bring more efficiencies, but as a also logical instrument, essentially a vehicle that can allow us to reclassify disease mechanistically. And that doesn't apply just to oncology, but to any therapeutic area where we can objectively make measurements, either through imaging or genomically or via biosensors and other objective mechanisms. Because if we think about precision medicine, this is something that inherently we've been, in a way, thinking about, even perhaps we haven't been articulating it in the context of AI and disease classification. But the underpinning of precision medicine is that if we give the right drug to the right patient at the right dose and at the right time, then that patient should respond. If we have a population of 100 patients, 100%, 100 of them should respond. That's the essence of precision medicine and an aspiration that many have been working towards in recent years. And unfortunately, most therapies do not work in all the patients that take those therapies. So if the response rate is then less than 100%, then one of these tenets of precision medicine must have been violated. And it's either the wrong drug for the right patient, or the right drug for the right patient but the wrong dose, or those could all be true, but then the therapy was given at the wrong time. What it turns out that the tenet that's violated the most is actually the right patient. We don't really have, using our standard diagnostic disease classification criteria, we cannot identify the right patients all the time. That's why we think about biomarkers. Biomarkers are a way to make our blunt diagnostic instruments or classification schemas more precise. So what's quite interesting is that a non-response is not bad luck. It's not a statistical anomaly. It's often a measurement and classification failure. And if we look at non-responders in any disease, but especially in oncology, where they tend to be what we call heterogeneous, is that the classification failure that's really the unspoken source of disease heterogeneity. If we had a very accurate way of diagnosing a patient, then there would be no heterogeneous population within that diagnostic classification. So the source really of disease heterogeneity is our failure in appropriately and accurately classify and diagnose patients. And that's reflected, in fact, in the response rates that we see in oncology. The more precise we are in our classification of a patient, the better the response rate. We're actually, in some cases, getting very close to having 100% of the patients respond. Let's start with non-small cell lung cancer before the advent of immunotherapies and targeted therapies. So in unselected patients in the days that we only had the microscope, which wasn't that long ago, before molecular profiling, the response rate to standard chemotherapy for a patient with quote unquote non-small cell lung cancer was about 20 to 30%. So one way to think about that is that our predictive accuracy of that diagnosis was quite poor, meaning the majority of the patients were misclassified because they just did not respond to the standard therapy for non-small cell lung cancer. Then came first generation tyrosine kinase inhibitors that were genomically selected. So EGFR is one of those that's present in 15% of patients with quote unquote non-small cell lung cancer. And the response rate in those patients is 40%. So we start to do better. What's interesting is that a non-small cell lung cancer patient with an EGFR mutation, we may call that disease something else. It doesn't have to be called non-small cell lung cancer. It's just an EGFR positive malignancy that happens to be in an organ that the surgeons used to call the lung, and we continue to do so today. And then tissue agnostic indications came to the scene. NTRK fusion is one. I already mentioned pembrolizumab, but NTRK fusions, patients with NTRK fusions have targeted therapies available for them, regardless of where the tumor is. The response rate is quite high, 75%. With second generation TKIs, we're getting very close to 90%. So the point of this response rate evolution is that the more precise we are in our classification of a disease, the better we can address that specific disease milieu, and the closer we get to this aspiration of precision medicine, right drug, right patient, right dose and time, to the point that 100% of the patients that we treat would respond. So in that context, imaging can in fact be used for patient classification to discover and surface the unknown patients subtypes and groups that we've been lumping together in these broad disease classification categories. And that a lot of people, and this is one of the things that I learned from Martin, in fact, is essentially the imaging phenome that we can start to think about the patient classification and diseases as they manifest in medical imaging as a phenotype, the imaging phenome. And this is not faster version of a standard measurement. This is not making your histological characterization more efficient or molecular profiling more efficient. The imaging phenome really speaks to a measurement that was previously not even possible for us to do. So this is where AI can go beyond ordinary automation. To really start to reclassify certain disease subtypes based on the imaging phenotype. And in many cases, these are imaging phenotypes and visual cues that are not visible to the human eye. So to recap, ground truth is what we call ground truth is often not truth. And our instruments are usually the problem, that we've had very noisy instruments typically emerging from human assessment, either visual or through other sensory input that is cognitively processed. And the AI models that have been trained on those labels inherit those deficiencies. So I'd like to show how flawed, as an example, how flawed our classification schema, which is the standard, the gold standard for approving drugs, is in oncology. So when I was at the FDA several years ago, we did a meta-analysis where we looked at registrational studies. These are studies that were submitted to the FDA and were approved for cancer therapies, where two radiologists were looking at the same image and then categorizing the tumor using the gold standard called the RECIST criteria. So it's a requirement that regulatory authorities have that in registrational studies, two radiologists have to review the images. So we looked at about 14,000 patients, and there was a 30% discordance, basically two different radiologists disagreeing more than 30% of the time. There were some interesting outliers. For example, one radiologist calling a scan a complete response, meaning they don't see any tumors, whereas the other radiologist says progressive disease. No, the disease is actually progressing. But overall, it was about 30%. But what's very interesting is that this was in 14,000 patients with all different tumor types. We started to look at tumor types. We did subgroup analyses and looking at tumor types like ovarian and pancreatic that are hard to measure. And the discordance was about 40 to 50%, almost reaching a coin flip. It actually gets worse than that because this was all based on RECIST classification. I'm not going to go into the details, the technical details, but RECIST is a classification schema that actually has a 50% buffer to account for human visual inspection errors. Basically, what that means is that we don't call anything response unless the tumor shrinks more than 30% from baseline. You don't call anything progression unless the tumor grows more than 20% from baseline. So that 20 plus 30% is a buffer where the tumor is certainly growing or shrinking. It's just not reliably visible to the human eye. So we call that stable disease. So a lot of drugs that operate in that 50% margin that unfortunately are not advanced because we can characterize the response and the dynamics of those tumors using human visual inspection. The majority of patients with metastatic disease unfortunately fall in that category where the disease dynamics, answering one simple question, is the tumor growing or shrinking, is actually very hard to answer. As Martin mentioned, I've been working and we've been working at Project Datasphere on an AI model development effort to automate the RECIST criteria and to take it a step beyond and develop an objective standard called Total Tumor Burden Index, basically an autonomous system that automatically detects the tumor and then volumetrically segments it and then reflects the total volume of the tumor burden in the patient. And we believe that's a much better reflection of tumor dynamics. What's interesting is that we're actually starting to see with our model a number of false positives. And in many cases, what's kind of interesting is that after adjudication, it turns out that our model is actually much more accurate than the original annotations. So a lot of the false positives we're starting to find out that end up being true malignant lesions that were missed by the radiologist. So the error is often in the human reference. And we're at the stage right now that image recognition models, we have hybrid models that are now incorporating transformer architectures with attention mechanisms, that these models are starting to rival what humans can visually detect. And this became a dilemma for us because we seems like we have saturated the reference standard, and we do not want to dilute our model and ignore the lesions and really be content with this false positive a reality that's starting to surface. So we have concluded that we can no longer use the radiologist annotations as the ground truth. It's too noisy, it's too volatile. We're getting a lot of false positives that turn out to be true lesions. We've thought about doing serial biopsies in patients, especially doing clinical validation of the model, but frankly, that's impractical. It's invasive. It's not fair to the patient. And what we are really going for, therefore, is benchmarking to survival. Essentially, as part of our clinical development program, since we cannot do serial biopsies and we have already surpassed what human visual inspection can do, we will be clinically validating the model in a prospective study benchmarking to survival. And what's interesting is that this is probably the best path forward for clinical AI, for a lot of AI models, where clinical validation is really the underpinning of not just regulatory approval, but adoption. And outcome validation is becoming more and more the price of entry into this domain. Now, there are some peripheral issues that have to be worked out. For example, a lot of technology ventures that are developing these models may not be used to conducting a prospective clinical trial. What I tell folks is that developing a diagnostic, an AI-enabled diagnostic software as a medical device, let's say, as the FDA calls it, in many cases, it's almost like developing an in vitro diagnostic assay or a drug. We have to show empirically that it works, even if we know all the constituents that are the characteristics of the therapy or the in vitro diagnostic. We have to empirically show that it improves real patient outcomes. So that is a completely different mindset than the augmentation mindset, which again, quite useful. We can augment human decision making. We can bring more consistency to how we treat patients, how we diagnose patients at a point of routine care. We can do things faster, but that by no means is discovery, revealing what we haven't seen yet. And that is really the act of discovery is what we need to advance science, to be able to improve longevity and bring better therapies to patients that need them. So clinical validation, as I mentioned, runs on prospective evidence, something that we all in the technology community have to reckon with and start to accept. And in many cases, that in fact requires a randomized clinical trial because there's so many unknown unknowns. These models are black boxes, just like many drugs are black boxes. I always tell people to randomly look at 10 FDA labels on the FDA website, look at the mechanism of action of that drug. In most cases, you'll probably see that mechanism of action is unknown. A lot of these drugs were not rationally designed. The rational drug design is relatively new, especially in oncology and now cardiovascular diseases with, for example, GLP-1s. But the majority of drugs have been approved based on screening assays. So the mechanism of disease are black boxes. The mechanism of disease is not really known. So we cannot administer these therapies and get them approved through mechanistic understanding alone. We've had to empirically show that they work. The same thing applies to AI. The best type of AI is actually black box AI with emergent properties. And in order to really advance those solutions and bring them to the market we need to empirically show that they improve important outcomes. So what I would like to propose is to replace the question that if a model is matching the gold standard with three different questions. And the first one being, how was the reference standard built and by which instrument? So if we're developing a model based on a reference standard with a volatile that was measured with a volatile instrument, that probably is not ground truth, and that bias and that imprecision will be inherited, and the model will essentially replicate it and scale it. How reproducible is the methodology independent of that reference? So this is what some people call technical validation. There are many different ways of approaching that. But the most valuable and the most important question, and perhaps the most challenging, is the third one. Does the evidence connect to something that patients really care about? Does my tool make a difference that is clinically meaningful? And can I measure that difference? And that is probably the best benchmark and ground truth to benchmark against because it is something that matters and it's something that can make a difference. And at the same time, it surfaces new insights that previously were not accessible to us. So in closing and in summary, I'd like to underscore again that there are no gold standards. In fact, there have never been gold standards. The entire engine of scientific progress and progress in medicine has been built on the foundation of questioning everything around us, including what we today call as gold standards. So no gold standards, only better questions to ask in ways that were never asked before. And I believe the work ahead is really hold these conventions, what we call gold standards, to higher standards and start to challenge them and build intelligent machines and clinical studies that are validated against clinically meaningful outcomes that really matter, not expert opinion or annotations that can be volatile. Because if we continue to benchmark against our latest vocabulary, a linguistic definition of what we observe, we are really bounded by the limits of what humans can do and observe, and we may not be able to leverage AI to its fullest potential, and we may forego accessing mechanistic insights and perhaps breakthrough groundbreaking discoveries and leave that for the future generations to tap into. So I think there's a window of opportunity today to use medical imaging, especially to surface these new insights, to start to understand what hasn't been visible to the human eye, to start to gain a better understanding of the reality of disease and in my case, oncological diseases, and really go beyond what we have been able to see and quantify, and really surfacing the unknowns. Thank you for your time.
Thank you so much, Sean. That was super, super, super interesting. A lot of new concepts, at least for me, many, many new concepts. So, you know, as an ophthalmologist, we do have that understanding that the signs that we see, for instance, in the retina, they are signs of systemic diseases. So we do have that understanding of like organ agnostic, more mechanistic formation of diseases. And that was something that was really established, for instance, when we started using anti-VEGF, such as bevacizumab, in the eye, and noticing the change that would make in a disease, and that is a drug that comes from oncology. So it's really that translational science across specialties that we've been applying. So that's super interesting to see that from the oncology perspective as well. So we have a few questions in the chat. So the first one is from Isabella, and she is asking if you could give us an example of an imaging-derived feature or phenotype that humans couldn't have defined on their own. And I would expand her question to imaging biomarkers that you think are relevant and perhaps were derived from AI experiments that you think are relevant in precision medicine.
Sure. The two categories of examples. One is that humans cannot measure reliably the volume of a tumor looking at a CT scan. And AI can. In fact, AI can do it autonomously. That's what we're doing with TTBI, Total Tumor Burden Index. And for radiologists, I guess technically it might be possible, but you would have to spend an entire day on one scan, measuring every single lesion and then calculate the volume. Nobody does that. And you may not be actually accurate because the boundaries of the tumor are sometimes the toughest to distinguish. So your volume calculations may not be correct. So that's one example, because what we're doing today, and what people have to realize is that patients in the metastatic setting, have multiple lesions all over their body. And what the radiologist is really, for those that have read radiology reports for patients with metastatic disease, you basically try to capture and describe as much as you see in the limited time that you have. And you focus on what's important. Maybe a lesion is close to an organ that could be dangerous, or a lesion that has grown substantially. So the radiology report is always, well, this lesion may have increased since last time. This one appears to be stable. And this one may be actually a little smaller. Clinically correlate. So at the end of every radiology report, there's that one line that essentially puts the obligation back on the treating physician, the oncologist. So the oncologist in the room is really looking at a radiology report as just one piece of evidence in treating patients. In most cases, they're going by how the patient is feeling and functioning. They're asking the patient question, how are you feeling? I can't get much out of the radiology report because there are multiple lesions. So the patient's signs and symptoms become quite important. In fact, the FDA used to accept clinical response and progression, essentially the physician's judgment as an endpoint for regulatory approval. So that clinician judgment drives the majority of treatment decisions today, not necessarily what's in the radiology report or on the scan. So that's one example, understanding tumor dynamics. We have a very two-dimensional reductionist ability to understand tumor dynamics. The best we can do is to give a general impression that maybe it's larger, maybe it's smaller, correlate with patient symptoms. The other category would be features that are not visible to the human eye. So I'm sure many people have heard about feature extraction. So what that really means is that you extract features of an object and you describe it as a cat versus a dog. Well, the same thing applies to tumors, that there are features that give you, that may look exactly the same to the human eye on the CT scan, but they would be mutually exclusive in the latent space of AI, meaning the trained AI model and its understanding of that image. And those features could correlate with mutations. There are very interesting efforts, published efforts, that have looked at tumor features that correlate mutations. So instead of doing genetic profiling on the tumor, sometimes it's very hard to actually biopsy those tumors, and you don't have much tissue. They say in oncology, they say the tissue is always the issue. So you cannot do molecular profiling, and the patient then may miss going on a targeted therapy. So you can, by detecting the features that are not visible to the human eye, you may be able to identify mutations. There's been a number of efforts that have been published in recent years. In the old days, we used to call that radiomics. And that's a field that actually has been around for a long time, basically trying to quantify the various features of what's seen on a scan. So a lot of the AI models now have supercharged that field, because you can imagine that quantification and that feature extraction is much more accessible to us today with AI models. So those are two categories of examples.
Yeah, that's great. You briefly mentioned FDA, and there's a question in the chat, and I also have one question of my own, so I'll try to combine them. So many of the companies that Segmed support, these companies are submitting their pre-market studies to the FDA and basically benchmarking their algorithms against human labelers. Do you think that is kind of the wrong approach, and how would these companies could do better? I know it's probably a regulatory requirement rather than on the company side. And Jayan from our audience is asking, what would it take for any of these AI-based imaging biomarkers or endpoints to be accepted by regulatory authorities as an endpoint?
Right. I think, well, that question is related to what you mentioned in a way, because the regulatory requirement and the whole benchmarking ecosystem as viewed through the lens of the regulator is quite interesting. As I mentioned during my presentation, that regulators have figured out a way to manage uncertainty in biomedicine and how we develop drugs and certain diagnostics through empirically. The controlled empiricism is really the backbone. In fact, it wasn't until last year where the FDA published, the former FDA commissioner in the New England Journal of Medicine, a new pathway that is completely mechanistically driven, that if you understand the mechanism of disease so well, that's no longer a black box. These are usually Mendelian diseases, genomically driven. Then we may be able to approve your therapy based on the experience of a handful of patients, and they give an example of an approval based on 6 patients. And then you just basically generate the evidence when the therapy is approved. So plausible mechanism, but that is probably 0.5% of diseases out there. So that managing uncertainty now, so the same thing applies to AI model development and digital biomarkers. Unfortunately, we hear a lot that nowadays that, oh, this AI software as medical device was approved. It turns out that these are not actually approvals. These are clearances. That goes back to what you were saying, that based on a predicate that is, again, based on human judgment, the 510(k) cleared. And unfortunately, nobody, clinicians, payers, guideline folks, don't really take that type of clearance seriously. And one may argue they shouldn't, because again, they're not saying it this way, but they're really based on labels that may not be ground truth. So what we really need is advance these solutions, AI-powered diagnostics, SAMDs, software as medical device solutions, and related devices and models through the PMA pathway, pre-market approval pathway, which requires in most cases a prospective study. And you let the data speak for itself, because if your model is PMA approved, your digital biomarker, let's say, It's a high bar. It requires conducting expensive clinical trials. But if it's approved, then naturally it will find its way into guidelines, and you don't really have to fight for adoption. It becomes the new standard. It replaces the old standard. That's how new therapies change practice. They become the new standard. Nobody's arguing for adoption, because if it's a new treatment for a patient that didn't have the treatment, well, that's the treatment that it should get. Same thing when it comes to AI model development. We haven't seen that yet. There are many different reasons for that. Again, part of it is lack of expertise in these companies. They need a clinical development team. Very few technology companies have a clinical development team internally. Their investors probably are still thinking that they're developing technology versus a medical solution or a therapeutic. They may not be comfortable with sort of that long R&D time that's required. But I encourage folks that are developing medical AI in cases where they actually want adoption and they want those tools to be used, especially if they wanted those tools to be used de novo, a tool that will bring something to a patient or a clinician that they didn't have before. Show it prospectively, because if the data is strong, you won't need to fight for adoption. It becomes a standard. It will be in guidelines. And that applies to essentially digital biomarkers.
Awesome. Well, Sean, I think I have a lot more questions. I know Aline has a lot more questions, but unfortunately we're already over time, so we'll have to close it out here. I think the main lesson here was there are no gold standards, which, by the way, is very much in line with when I started a master's in epidemiology years ago. The first lesson we had there was never use the word gold standards because they don't exist. Use reference standard instead. But anyway, it's interesting that it comes back here now. So Sean, I really want to thank you for your contributions and excellent presentation and great discussion afterwards. So thanks for being here. I would also like to thank Aline, my co-moderator, and Andrea, who the people here don't see, but she's behind the scenes organizing everything. And of course, the audience for coming up and showing up and asking some really good questions that resulted in a nice discussion. The next Bytes of Innovation webinar will be on Tuesday, July 7th with Nijat Ahmadov from Nux AI. So I hope to see you all then. And thanks again, Sean.
Thank you.